Oktober 2019, RSAB harapan kita ditetapkan oleh Profesor Dr. Nilafari Amfasamuluk, SPN, sebagai Pusat Kesehatan Ibu dan Anak dengan nasional dengan Surat Keputusan Kesehatan Republik Indonesia. Sesuai dengan visi RSAB harapan kita, yaitu terdepan dalam pelayanan kesehatan perempuan, perinatal, dan anak RSAB, harapan kita berperan penting untuk mengembangkan layanan kesehatan melalui berbagai riset berbasis klinik. Saat ini kasus-kasus rujukan yang akan dihadapi PKIA akan semakin banyak dan kompleks sehingga membutuhkan peningkatan kemampuan dan keterampilan klinis sumber daya manusia selain sarana dan prasarana. Saya harap dengan kegiatan ini kita semua sama-sama belajar dan dapat menerapkan ilmu yang didapatkan serta bermanfaat bagi seluruh peserta dan kita semua. Waalaikumsalam warahmatullahi wabarakatuh.
Thank you so much, Dr. Vivianti, for the opening messages. Now, to also welcome us, please welcome Dr. Winda Aswanti to the stage. Dr. Winda.
Thank you very much, Dr. Vivi, Pak Basari. I'm Dr. Winda. I'm pediatric cardiologist from National Women and Children Hospital Harapan Kita. Please let me welcome Dr. Justin Daniset and Dr. Vishnu Vasantan from University of Calgary. I believe both of you are in the Libyan Cardiovascular Research Institute. We want to say hello to both of you. Thank you so much to accept our invitation to this guest lecture. And we have so many participants this morning. There are medical students, biomedical engineering students, as well as the expert in biomedical engineering at Faculty of Engineering, Universitas Indonesia, and also from Faculty of Medicine, Universitas Indonesia. Thank you so much for coming to this virtual meeting.
Thank you so much, Dr. Winda. Now, I bet that everyone is excited to finally start off our event. Without further ado, I would like to first introduce our moderator for today, Dr. Winda Aswanti. She is a pediatric cardiologist at the National Women and Children Hospital Harapan Kita. For Dr. Winda CV.
Thank you, Yasna. Ladies and gentlemen, dear students, lecturer, professors, and of course our distinguished guests from Libyan Cardiovascular Institute, Cumming School of Medicine, University of Calgary. Let me be your moderator this morning or this evening in Calgary. We have two speakers today. I would like to give the first talk to Dr. Puspita Akatili. She's a lecturer at the Biomedical Engineering Study Program at Faculty of Engineering, Universitas Indonesia. Would somebody help to show the curriculum vitae of Dr. Puspita? Dr. Puspita has finished her PhD program at University of Nottingham with Chris Denning. And actually, she is a medical doctor, and she continued her education at stem cell technology at the University of Nottingham in 2014. Continued to obtain her doctoral degree at the same university, and I think she has very big interest in cardiomyocyte and especially of the human-induced pluripotent stem cell. Without further ado, I will let Dr. Puspita to give her talk. Please, thank you.
Okay, thank you very much, Dr. Winda. Can this screen be stopped so I can share the screens? Thank you. Okay, good morning as well as good evening. So this talk is more like an introduction because it gives you a general background about cardiac tissue engineering and also the application, such as particularly focusing on engineered heart tissue in drug screening, disease modeling, and regenerative medicine. So I will first talk about the like what is iPSC cardiomyocyte, and then especially the counterpart itself, the embryonic stem cells, and then examples of the cardiac tissue engineering and the approaches. And then the last one is the applications, particularly engineered heart tissue in many applications. So these are the abbreviations used in the next couple of slides. So why we bother with a human model? So all this time we've been using animal model, but unfortunately there are some differences, especially in the physiology, because for animal models, we start with the small models, for example, the mouse cardiomyocytes. So there are about 20 different of protein coding genes between mouse and human. And then also about the nucleus. So the mouse cardiomyocyte mostly have binucleated, while the human are mostly polyploid nucleated. And then for the myosin heavy chain, which is one of the sarcomeric structures, so they have different expression. That's why it differs in the contractility function between mouse and human. And of course, the electrophysiology, the expression of the ion channels as well. So that's why we need a human model in this one. And then especially there, since the development of the induced pluripotent stem cell, there's fast studies about making a human platform. So as we all know, so basically for pluripotent stem cells, there are two sources. First is derived from the inner cell mass of the blastocyst, which is embryonic stem cells. And then the recent one is the popular because it's derived from somatic cells. For example, in here is fibroblasts. So using the Yamanaka factors, we created induced pluripotent stem cells. So both of these pluripotent stem cells then differentiated into cardiomyocytes. But what we've been generated so far, they have immature phenotype. So it's more like in about 20 to 14 weeks of the gestation period. So that's why we need to mature them into reaching an adult state. But is it as simple as that? No, that's the problem. So that's why we need to bioengineer to mimic the human heart tissue. And the approaches, we'll talk about it in the next couple of slides. So these are the characteristic properties between immature cardiomyocyte and mature cardiomyocyte. So from the structural to the functional properties. So the distinguish from the structural is the sarcomere. So in immature, you can see the pattern. The Z bands are immature, irregular, and with mature, you can see it's highly organized. And then the calcium handling as well. And for example, ion channel, gap junction. So gap junction, for example, is connexin 43. In immature, it's more circumferential, but in mature, it's more concentrated at the intercalated disc. And for metabolism, immature is using glycolytic, while as mature is using oxidative phosphorylation, and so on. You can see it in here later. Okay, so here are the key measurements for the maturity. So for the contractile force, because the mature one generates high force and faster upstroke and decay. And this here also responds to adrenergic stimulation. So it's responsive to inotropic drugs, which I will show you later in the recent study. And then this is also important, increased force reconciliation shift. So when you pace them, the faster you pace them, you should see an increased force. So it's showing a positive staircase. And then conduction velocity, transcriptome, and metabolism. So switch from glycolysis to fatty acid metabolism. So this picture shows you like a general maturation protocol. So not only cardiac tissue engineering, but also a more simpler approach. I'll start from the right side. So in this one, so the cardiomyocytes, they are injected into the rat. So they are tagged with green fluorescent protein to differentiate from the red cells in the red. So they are like some kind of like incubation in the mice. So they are expected to be mature. And then fatty acids. So that's why because like metabolism, when they are mature, they are using fatty acids. So hopefully by using this, they will become more mature. And then co-culture. So using another cell as well. The most popular one is the endothelial cells and fibroblasts, because they are the cells that are often found together in the cardiovascular tissue environment. And then these are the combination of chemical factors and metrogeometries. So in here is hormone T3 and then glucocorticoid. And then these are the one of the cardiac tissue engineering approach. So using a 3D, this one is using electromechanical stimulation. So increase spacing from 2 Hertz to 6 Hertz. And then this one is just a 3D cardiomyocyte without using any stimulation. So these are examples of many cardiac tissue approach. So by using the based on the factors like a substrate, growth factors, mechanical stress, static stretch, and then biomaterials. So we are combining these factors to create a cardiac tissue engineering. So the most simple one, the simplest one is the cardiac spheroids. So basically because it's made from aggregates. And then this is the EHG. So it's from 2 post-planar or ring shape, which I will focus on later. And then this is example of combined EHD and biowire, and then MPS, and then 3D bioprinting and so on. So these are the examples, like basically the scaffold categorized based on scaffold-free or scaffold-based cardiac tissue engineering. So for example, the microtissue, 3D, and then cardiac sheet is scaffold-free. So these are how they are fabricated. And then the scaffold base, for example, muscular thin films, EHD, of course, cardiac wise and hard on chip. They are scaffold base. And then some of the cardiac tissue engineering approaches can be made for regeneration approach by either transplantation, transdifferentiation, cell proliferation, and then also for gene therapy and drug testing. Okay, so this one is about the application of the cardiac tissue engineering. So the ultimate goal of the cardiac tissue engineering is, of course, the cardiac regeneration. So using some kind of cardiac patch, so you place them in an infarcted region. And then also, for example, the hydrogels for the increased survival. And then also for disease modeling, so we can know the pathogenesis of the disease. And then also for drug screening, so helping in either like discover a new drug, or for to prevent the economic loss, because currently in the pharmaceutical industry, most of the drugs developed cannot reach phase 2 clinical trials. Okay, so how to choose? So depending of what you need, either drug screening, regenerative medicine, and disease modeling. So, and depending on the cell type also, and then the assay. So EHD here is symbolized in blue color. So you can see EHD can be used in every assay: structural analysis, single cell electrophysiology, and then contraction force, which is very important because one of the key maturity is the contraction force, and then the conduction velocity. And these are the advantages of each cardiac tissue engineering. As you can see here, EHD is the only one that can be used for drug testing. This is modeling transplantation. And then with advantages, because they can directly measure the force contraction, and then can be miniaturized and can be scaled as well to medium and high throughput. Okay, so first application is for drug screening, of course, because in developing a new drug, like they can be about 100,000 compounds already. So usually the first phase is through in silico, so with computer. And then after that, maybe around 1,000 is selected for the clinical studies in vitro setting, so in the lab. So this is the important function of the human model in here, iPSC, in screening the compound which is best, and then go through the phase 1, phase 2, and phase 3 clinical studies. And then after approval, go to the phase 4. So this is our study that was published back in 2020. So this was a collaboration between 4 universities. So University of Nottingham, and then University of Glasgow, University Leiden, and then Hamburg University, as well as a pharmaceutical company, GlaxoSmithKline. So in this study, we compared the 2D platform versus 3D platform in predicting the inotrope drug response. We also did electrophysiology and calcium handling study as well, but because not many papers established the contractility, in this paper, we focus on the contractility aspects. So in here, so there are positive inotropes. So positive inotrope is the contractility drug that affects contractility, which increase force contraction. Negative inotrope, of course, is the vice versa, and the neutral effect, so without effect. And as you can see here, we have the control, which is isolated rabbit cardiomyocyte, which is considered control for adult cardiomyocyte. And we chose rabbit because rabbit exhibit a positive staircase effect in reverse with the mouse, because mouse cardiomyocyte have negative staircase effect. And then for the 2D platform, we have a commercial cell line, which is pluricyte, and then iCell square, and then our own homemade cell line rebel pad. And then for the ERC, it is the cell line from Hamburg for the 3D. So in here, you can see like even in rabbit cardiomyocyte, it's not as good as the EHT here in predicting the positive inotrope, while for the negative inotrope and neutral effect, most of the platform can predict well. So how we can improve the prediction for positive inotrope? So one of them is by slowing the baseline bit rate. So with using ifabradine, we managed to show that it shows sensitivity, so it's more significant in detecting most of the positive inotrope drugs here. This is using the EHT platform, so you can see they are showing significant statistic. And then the second one is for the disease modeling approach. So in here, this is the why we are using iPSC, because iPSC carries the genetic information from the patient. So for example, patient from the skin fibroblast, and then you reprogram it, and you can get the patient specific IPS. And then for example, also the normal IPS from the patient who are healthy. And then you want to do the genome editing, you want to induce the mutant IPS, and then you differentiate them into cardiomyocyte, and then you can do these assays. So why we are doing this? Because we want to create a perfect control cell line. So for example, in here, the normal IPS, you genome editing them to introduce disease variants, or from the patient who already got the disease, and then you want to correct them into the normal IPS, so we can compare them each side by side, because it's a perfect control. So the only difference is the mutations. Okay, so this is because we are in a pandemic era, so I want to show you the application of the EHT in the COVID because it models the myocarditis found in COVID patient. So in here, so mock is the control because it only includes the empty factor, so there are no virus inside. So by the immunohistological staining, you can see here there is a white staining. So it shows the viral nucleocapsid protein, so from the virus. So you can see it shows in the COVID EHD compared to the mock. And then this one, the COVID gene. So mock here is the one with the 0 thing. And then the one with the COVID infection shows a high gene expression. And this one also, this is showing many cellular with the spike sense and antisense in the in situ hybridization. And then this one is the contractility profile. So it shows that compared to mock, the COVID infected EHD shows reduced contraction time, and then reduced relaxation time. And then this one also showing the cardiomyocyte death with a tunnel. And then this one showing the sarcomeric disorganization with the troponin T. And then a wider aspect of the remdesivir application showing the phenotype of the COVID infected EHD. Okay, so this is the last one. So this is from the injured heart, so for regenerative medicine application with EHD. So this is the contractility profile. And then this one is, so they made 2 versions. So this is the normal patch EHD, and this is the smaller version of EHD. And this is left ventricular, and then it shows that the patch EHD version match more to the left ventricular compared to the small EHD. And this is the electrophysiology profile, also showing that the patch EHD is more showing matching with the left ventricular side. And then this one shows, so they have 3 different doses for the EHD, and then they made the Guinea pig here with infarct. And then they want to see, this is the slice from the base to the apex, and then compared to the without the patch, and then the first dose, second dose, third dose. So the one with the asterisk here is the one with the human patch here. And then they want to show the scar size, and then the cell fractional area change from post-injury to the final. So they want to see the improvement of the regeneration using these 3 different doses. Okay, so the challenges are because the EHD needs to be scaled for different applications. So the ones for drug screening and the one for disease modeling and regenerative, of course, may need different version of EHD. And then because EHD requires a very large of cardiomyocyte, of course, there's the cost versus benefit. And then impact on the GMP, but a recent study shows that it's already GMP compatible. And then the increase of the quality control, obviously. And then for the higher throughput system, it needs a further modification. And then because the AHD, because they have smaller and large version, the large version needs improvement in the supply of the oxygen and nutrition to avoid necrotic area. But recent studies have shown to improve this. Okay, so to summarize, despite they are still amateurs, the HPSC cardiomyocyte, they are still very useful in drug screening, disease modeling, and cell therapy. So we may not need adult HPSC cardiomyocyte for every settings. For example, for cell therapy, because we want the cardiomyocyte to proliferate, while in a more mature stage, the cardiomyocytes are more quiescent, so we may need relatively immature. But we need the adult cardiomyocytes to model particular disease, especially related to adult stage, for example, metabolic syndrome. So we need investigation for multiple approach in mechanical loading, pacing, metabolic substrate, and cellular composition to drive the maturation for EHD, which is very much needed. Okay, so here are the references. Thank you.
Okay, thank you. Thank you so much, Dr. Puspita. My opinion, you have give a very interesting lecture regarding the basic of human iPSC, and it's useful. I think we will proceed to Professor Fedak lecture. Professor Fedak is the director of Libin Cardiovascular Research Institute in Calgary, Canada. We are really happy that he could accept our invitation to our guest lecture. Prof. Fedak also accompanied by Dr. Vishnu Vasantan and Dr. Justin Denisat. We are really happy to have all of you here. And let me show your curriculum vitae, Prof. Fedak, because I want to, everybody wants to know you. And after that, we will proceed to your talk. So Prof. Fedak is a professor of cardiac sciences. Now he's the director of Libin Cardiovascular Institute. He's a cardiac surgeon, but he is his interest also in clinical scientists in cardiac paths, and we could search for his publication. So many publications, and I think now is the time for him to give his talk. Please, Professor Fedak.
Great, thank you very much. Can you hear me okay?
Well, very good, yes.
Excellent. Okay, well, good morning, everybody. It's evening here in Canada. We're on the west part of Canada in the Rocky Mountains in Calgary. I'm really excited to engage with all of you so far away. And we're really grateful to be invited to speak with you today. So thank you very much. I would like to let you know that there are two other speakers involved in this talk, because I'd like to take you through some of our work. And of course, like many of you, we work in teams. Dr. Vishnu Vasantan is a medical doctor and a cardiac surgery trainee who's been working on his PhD for many years in my laboratory program, and you'll hear from him throughout this talk. And then you're also going to hear from Dr. Justin Denisat, who is an assistant professor in our department, and he has some really exciting expertise and work in the area of immune cells and the cardiovascular system, particularly in the pericardium. And you'll see how we're all working together to try to advance cardiac repair and regeneration. So Vishnu, why don't you go to the first slide? So I'll get started, and then I'm going to pass the microphone over to my colleagues. Next slide. Okay, so my journey into exploring cardiac repair and cardiac regeneration has been over a couple of decades. Back when I did my PhD in Toronto, there was a lot of excitement about cell therapy. So we believed very strongly that we could take cells like stem cells and other cells and inject them directly into the injured heart, and that they would regenerate and repair the heart. And we've learned over time that it's more challenging than we thought it would be. And throughout my work, there was so much attention on cardiomyocytes and the cells that it was almost that people were forgetting about the healing process. And a lot of my work has been focused more on the healing process to see, can we alter the healing process in the heart? Can we make the heart heal in a different way? Because as many of you know, the heart is really unique as an organ in that it really scars down. It creates a lot of fibrosis after injury, and that fibrosis leads to a loss of function. And we all know as clinicians, the more heart function you lose, the worse you do over time and the worse your clinical outcomes. So if we had a way to get the heart to be less fibrogenic and maybe more adaptive in its repair, more vasculogenic. So that has been my interest for quite some time. So in many ways, it's not true regeneration. I'm not trying to take scar tissue and turn it into functional muscle. That is really exciting technology. And the talk that we just heard was excellent. And I think that iPS cells are really the future, and there is going to be regeneration at some point. But my work is trying to take a little bit step back from regeneration and say, can we do something a little bit simpler that may not take as long to develop, something maybe we can be doing right now for our patients that really directs a repair process that's not so fibrotic and is a little bit more adaptive to restore function. And I'm going to show you some of our data today about how we've been working on that. So as we know, cardiac fibrosis is really a problem, and we've really focused a lot of our attention not on the cardiomyocytes, but on the cardiac fibroblasts, because they are the big mediators of the repair process. There's really two main cell types that we know affect repair: cardiac fibroblasts and the immune and inflammatory cells. And that's why Dr. Deniset's work's been really critical for us. We're joining forces to better understand both of these cell types in the healing of heart muscle after ischemic injury to see if we can alter it. Next slide, please. And the way that we've approached this is by looking at biological extracellular matrix sheets and materials. So I've long believed that if we could find a way to repair the heart and redirect the healing process to be more adaptive and more functional without using stem cells and using the innate healing processes within the heart, or maybe mobilizing the stem cells within the heart itself, that we would have a potential therapy that was a little easier to develop. We could develop it faster and we could get it into patients more easily. And I've been really interested in different biomaterials, and I'm really interested in biologic extracellular matrix biomaterials because, as many of us understand and know, this matrix material in the human tissues really drives what the cells do. So my thought was, if we take matrix sheets that have biologic signals and have bioinductive effects, if we put that on the heart or in the area of injury, maybe we could influence the reparative cells to function in a different way and drive that adaptive repair process. And one of the materials that I've been really interested in is this one called Cis Ecm. It comes from the porcine small intestine, and it's really the basement membrane of it. And it's decellularized, so it retains the three-dimensional matrix structure, it retains the signaling properties, but it doesn't have the cells that could actually interact and create more inflammation or be rejected in human tissue for application. Next slide. So our journey with this work was really trying to take these bioinductive and bioactive matrix materials and placing them directly on the surface of the heart after injury. Since I'm a cardiac surgeon, I deal with an open chest and I deal with patients who have acute MI, and I go in and I revascularize them with bypass grafts. And it's always struck me that we're missing an opportunity every time we're in the operating room after an acute injury, is that we just ignore the muscle. We do nothing for it. We create inflow with the bypasses, but we don't address that muscle. And we know that muscle is injured. And over the coming weeks and months, it's going to continually remodel, it's going to thin, and it's going to get fibrotic. And maybe there's something we can do in the operating room at the time. And the idea was maybe we can take these matrix sheets, place them on the epicardial surface, and that would create the signaling that we would need to change the course of the healing process. So we put that to the test. And some of the first studies we did was in rat models. And we showed in a rat model that when we applied this patch therapy, we actually were able to improve heart function. And we saw changes in fibrosis, and we saw more vasculogenesis after we placed this patch. So we did another test after that, and we inactivated the scaffold. We said, well, is this really a biologic signal, or is it just a biomechanical effect? Are we just restraining the infarct and that's why it's healing better. So you can see in the other graph there where it says inactivated scaffold, we did a chemical treatment on the scaffold and then placed it and saw that it did not protect the recovery of function in the same way. So this was important information for us to know that it is the biologic dynamic signaling of this scaffold that is driving the reparative response. So we did a lot of other work to try to better understand why that was happening, but we did want to take this down the translational pathway. And you saw in the first presentation, you know, the very nice translational pathway where you go from the bench to small animal models to a preclinical large animal model, which is typically a pig. So we did exactly that. We created ischemic injuries in a pig model and we placed the patch on the pig heart. We then sent those pigs into the MRI scanner and evaluated their heart function and their healing. And we looked at the tissues themselves. And we saw a remarkable improvement when we placed that patch very early after the infarction. And interestingly, by MRI, what really surprised me was by clinical measures, those areas were considered non-viable, okay? And yet when we put this material, the patch material on it, we actually saw a restoration of function in what would be clinically called non-viable myocardium. And that to me is really important because it suggests that many of our patients that we see in clinical practice who have so-called non-viable areas of injury actually could be restored with the right kind of signals and therapies. So it's a huge opportunity to treat patients. Next slide. So then after doing the preclinical work, of course, we needed to test this in human patients. So we've done an 8-patient study, just a very small study, not randomized, feasibility, where we mimicked what we saw in the pig model. And we took patients who had acute ischemic injuries, we put them in the MRI scanner, we identified where the ischemic injury was and the extent of the injury. And then when they went to cardiac bypass surgery, within about a week or two after their infarction, we targeted the area of injury by placing the biomaterial on the epicardial surface. Really simple. This is out of the box technology. It's decellularized. It's actually already FDA approved. So this product exists on the market. So we're able to easily do the study. And we basically cut it out, sewed it on, took an extra 5 minutes on the surgery on the heart. And then we looked over time at 6 weeks and 6 months after to evaluate what happened in these areas. Next slide. And remember, this is not randomized, but what we did was we looked in the areas that we bypassed and we looked at the areas where we placed the scaffold and the bypass. And here's 2 examples of patients where we saw in the areas that we added the scaffold and a bypass, we saw a much greater result in the regional myocardial perfusion relative to the overall perfusion of the heart, really strongly suggesting that in these human patients, the scaffold was able to do what we thought it could do, and that's drive a vasculogenesis and angiogenic response that could result in improved function. Now, we still need to take this further, and we would like to test this in a randomized trial, and we're designing that now and hoping to go forward with it. So this epicardial patching therapy with a decellularized material is a very clinically feasible, something that we could be doing right now to improve our patients. And if you think about it, when we do bypasses, we provide inflow, but we don't actually address the microvascular damage after an infarction. So we address the macrovascular damage and we provide inflow, but we really need to get the microvasculature fixed as well and reduce the fibrosis and scarring. And I believe that this potential therapy can do that. Next slide. So we've tried to better understand as well how this could be happening. And we do believe that the material is altering what the fibroblasts do. So I talked about those 2 important cells in the healing process, the fibroblasts and the immune cells or inflammatory cells. And we extensively looked at human cardiac fibroblasts and we interacted them with the material. And then we did some analysis to see what genes were being changed. And When we did this, we found very interestingly that the fibroblasts themselves seem to be redirected towards an anti-fibrotic response and an increased vasculogenic response, just like what we expected and wanted to see. So we saw markers of angiogenesis very much increased, and we saw markers of fibrosis decreased when the cells interacted with the material. And remember, these are human cardiac fibroblasts from the types of patients who have ischemic injuries that we'd want to treat. Next slide. So I'm going to stop there and I'm going to pass it on to Dr. Vasanthan, and he's going to tell you about more of his work, how he's extended these observations, and we're going to tell you a little bit more about how we've developed this therapy to be more applicable to many, many more patients than just those who go to the operating room. So Vishnu, why don't you take over?
Sounds great. Thanks very much, Dr. Fidak. Firstly, can everybody hear me well?
Yes, very clear.
Perfect, thank you. As Dr. Fidak so kindly mentioned, my name is Vishnu Vasanthan, and I'm a cardiac surgery resident at the University of Calgary, and I'm also a proud member of his lab. So over the last few years, we've been investigating new angles for cardiac repair using biomaterials. As Dr. Fidak had mentioned, we've investigated modulating the cardiac fibroblast from an angiogenic or from a fibrotic phenotype to an angiogenic phenotype. And with the study that I'll be presenting today, we look at a new angle for cardiac repair. This was an article that was recently published in the Journal of Thoracic and Cardiovascular Surgery. And not only does it look at fibrosis and angiogenesis, but the main focus here is how the biomaterial can be used to augment cardiac inflammation to promote cardiac repair. So the central message of this talk is that we applied an acellular matrix biomaterial, so Cis Ecm, to the surface of the heart or the epicardial surface after myocardial infarction. And we investigated how the biomaterial impacted cardiac inflammation. The biomaterial we found redirects fibroblast paracrine activity to release inflammatory cytokines and angiogenic factors. And by doing so, it recruits pro-angiogenic immune cells and subsequently increases neovascularization. Now, this slide was previously shown, but it serves as the preliminary data that justified this study. So as Dr. Fidak had mentioned, this RNA sequencing data of cardiac fibroblasts seeded on Cis Ecm show that the fibroblasts change their transcriptional activity. They become more angiogenic and less fibrotic. Now, I reanalyzed this data set at the start of my thesis and also identified another angle in which the fibroblast changes its activity. Along with its angiogenic and fibrotic changes, we found that there is an increase in the transcription of angiogenic genes. For various themes, there were increases in the general inflammatory response and the recruitment of specific immune cell populations. And with this in mind, we asked the question of, could this be one of the other mechanisms that are impacting cardiac repair with this biomaterial Cis Ecm? So the objective of this study was to explore how Cis Ecm modulates myocardial post-infarct inflammation, and how does this impact cardiac repair? Now, the main model used in the study was a mouse infarct model, and there were two procedures performed in the same sitting. The first was a coronary ligation. So the coronary artery is ligated to surgically induce the infarction. And in the same procedure, the biomaterial Cis Ecm is applied over the infarcted area. And from there, we could do many different types of studies, including myocardial imaging and flow cytometry. Now, one of the first studies we did was 2-photon microscopy in this mouse model. So in this 2-photon microscopy image, we can see the biomaterial seen in gray over the myocardium. So the biomaterial is implanted at this point. And this is 7 days after the myocardial infarction and the procedure. Now, fibroblasts in this image are labeled in red. And as we zoom in, we can see that after 7 days, the biomaterial is taking in fibroblasts from its surrounding. And this is a very interesting finding for us because it identifies a point at which the biomaterial takes in these cells and provides an opportunity to modulate cellular activity. In a sense, the biomaterial, by taking in cells from its surroundings, is creating a new construct where it interacts with these cells to create a bioactive mechanism. Now, to investigate this biomaterial after it's been embedded, we explanted it and then cultured it ex vivo. So the biomaterial was taken and placed in cell culture media for 24 hours, and this allowed it to elute various factors that it was producing. And it was compared to an unimplanted, non-embedded control. And again, this is Cis Ecm in all groups. And what we found is after embedding the biomaterial on the heart, it started to produce VEGF, which is an angiogenic molecule, and inflammatory cytokines like keratinocyte chemoattractant, granulocyte colony stimulating factor, interferon gamma induced protein 10, and interleukin 6. And these inflammatory cytokines are related to the recruitment of monocytes, neutrophils, and a general inflammatory response. So taken together, after embedding this material on the heart, it becomes able to produce factors that induce an angiogenic and inflammatory response. Now, given that we labeled fibroblasts earlier, we wanted to see if fibroblasts are one of the cell types that are producing this inflammatory and angiogenic response. So we seeded mouse 3T3 cell line fibroblasts on the Cis Ecm biomaterial and evaluated their protein production when compared to a control group, where the fibroblasts were seeded on an inactive control. And what we found is that the fibroblasts seeded on the active biomaterial had that pro-angiogenic and pro-inflammatory response as well, linking these cells to the response that was seen in the biomaterial. Now, after understanding that fibroblasts are involved in this mechanism, we wanted to identify how the biomaterial is changing fibroblast activity. And from here, we identified different components that can potentially change fibroblast activity to promote this response. Fibroblast growth factor 2 was previously identified in our lab to modulate fibroblast activity. And in this slide, we show that the biomaterial is releasing fibroblast growth factor 2, so it still contains this factor. Additionally, we performed 2 photon microscopy on the biomaterial itself and used a nuclear stain. The biomaterial is acellular, but the decellularization process seems to leave behind nuclear material. And this is residual material left behind by cells that used to live in the ECM. So what we see here is residual nuclear material and fibroblast growth factor 2. And both of these can potentially change cellular activity. So from here, we performed an inhibitor study to further understand the impact of these mechanisms on fibroblast activity. So the main point of this study is we took the bioinductive factors from Cis Ecm and exposed 2D cultures of mouse fibroblasts to those bioinductive factors. And we added different inhibitors. So one for fibroblast growth factor 2 and another for DNA sensing toll-like receptor 9. And we wanted to evaluate how that changes fibroblast paracrine activity. And in terms of VEGF, the main summary of this somewhat busy slide is that fibroblast growth factor 2 and toll-like receptor 9, the DNA sensing mechanism, both potentiate VEGF production from fibroblasts. And then when we look at the inflammatory response or the inflammatory cytokines, we see that the cytokine production, depending on the individual cytokine, is mediated by FGF2 alone or toll-like receptor 9 alone, or in some cases both. So we start to understand the mechanism of how the fibroblasts are changing their activity. Next, we ask the question, so what? So how does changing fibroblast activity impact the myocardial inflammatory process after myocardial infarction? And here we went back to our mouse model. There were 2 treatment groups in this case. Both treatment groups received the myocardial infarction. And there was a control group with a sham surgery, and the treatment group had Cis Ecm implanted. And then flow cytometry of the myocardium was performed at 3 and 7 days. And from the study, if you look at the panel on the left, we see the myocardial flow cytometry results. And at day 7, we see a sustained inflammatory response in terms of early responders to myocardial infarction, so neutrophils and inflammatory Ly6E-high monocytes. This suggests that likely because of the pro-inflammatory cytokines that are being released, there is a sustained inflammatory or early inflammatory response after the myocardial infarction at day 7. And furthermore, interestingly, Cis Ecm seemed to recruit these immune cells into its structure as well. And now, along with the general inflammatory response, we also wanted to investigate specific reparative immune cell populations. And for that, we turn to the VEGFR1 positive proangiogenic neutrophils and monocytes. So VEGFR1 positive neutrophils and monocytes have been shown in the literature to help with angiogenesis and new blood vessel network formation in ischemic muscle. And in this case, after adding the Cis Ecm or in the treatment group, there was a heightened response from the VEGFR1 positive neutrophils and monocytes at day 7. Given that the fibroblasts are producing VEGF and also inflammatory cytokines, we believe it's likely that these cytokines and VEGF are creating the appropriate environment to facilitate the recruitment of these reparative immune cells. And interestingly, again, Cis Ecm is recruiting these immune cells also into its own structure as well. And from here, finally, we also compared the proportion of VEGFR1 positive reparative immune cells in the myocardium when compared to Cis Ecm. And interestingly, for both neutrophils and monocytes, the Cis Ecm biomaterial contained a higher proportion of VEGFR1 positive reparative immune cells. The reason that this is important is that we believe that the biomaterial is creating a concentration gradient to facilitate that recruitment of these reparative immune cells. And as we look into this, we believe that the myocardium is benefiting from this concentration gradient that is being created. So now with a heightened inflammatory response and a proangiogenic angle to this response, we then asked, how does this impact the myocardium? So for here, we turn to resonant confocal microscopy. Essentially, this is high-resolution confocal microscopy that takes multiple small high-resolution images and stitches them together to form a larger image of the myocardium. And one of the main stains we used is isolectin. So isolectin is a small blood vessel stain. So it stains small blood vessels that can be seen in the microvasculature level. And here we do see isolectin or small blood vessel signal enhancement near the biomaterial. And it suggests that the biomaterial is inducing a regional angiogenic response that could potentially be reparative and be an underlying mechanism for the cardiac repair we've seen in previous studies from our lab. And furthermore, we wanted to quantify this response. And to do so, we compared the myocardium that was within 100 micrometers of the Cis Ecm material to the myocardium that was further than 100 micrometers. And it seemed that the myocardium closer to the biomaterial had a higher isolectin signal or small vessel density. And this likely signifies a regional neovascularization response that is localized to the biomaterial. So to summarize this study and go through the underlying mechanism, from our first investigations, we found that fibroblasts migrate from the myocardium into the Cis Ecm biomaterial. And this provides Cis Ecm with an opportunity to modulate the fibroblast secretome. This is through fibroblast growth factor 2 and also toll-like receptor 9 mechanisms to modulate how the fibroblast releases proteins. And we found that there are pro-inflammatory cytokines released and also pro-angiogenic VEGF. And these cytokines and VEGF create the perfect environment to facilitate recruitment of pro-angiogenic neutrophils and monocytes. And with the VEGF itself and these pro-angiogenic immune cells, we believe this is mediating the neovascularization we see after implanting this biomaterial. And the reason this is important is that toll-like receptor 9, as well as fibroblast growth factor 2, are noted to potentiate angiogenesis. And we've identified for the first time that the biomaterial can leverage, especially toll-like receptor 9, along with fibroblast growth factor 2, to potentiate repair by modulating cardiac inflammation. And we hope to leverage these responses to further augment these biomaterials in future studies. And with that, thank you very much for your time. And I'd like to pass everyone's attention over to Dr. Justin Daniset.
Thank you, Dr. Vasantan. I think Dr. Daniset can start your lecture.
So Vishnu, I think you have to unshare.
Is it still sharing or?
It is, yeah.
Okay, sorry. Here you go.
All right, so thank you, Vishnu, for sort of introducing some of what we're going to talk about, and basically trying to expand on this idea of communication from outside of the heart into the heart to actually regulate remodeling. And what I'm hoping to sort of take you through is the idea that the pericardium that surrounds the heart is actually very important for this process. And I'll just note that Dr. Verzanten just told you about the Cis Ecm, which is pictured here in green, that sits on the epicardial surface. But essentially, this biomaterial is placed into, quote unquote, the pericardial space. And so the question is what are the factors and cells within the space that may also play an endogenous role in the native healing response? And because I come from a more of an immunology sort of mindset, my interest, as Paul had mentioned, in addition to fibroblasts, is immune cells play key roles in the remodeling process after injury. And now for a long period of time, this recruitment or the cells that get to the site of injury were thought to uniquely come from the bloodstream. And so here, just in a normal beating mouse heart, we can visualize particularly neutrophils and monocytes that circulate continuously through the capillary beds of the heart. But soon after you induce even just a small injury is that many of these cells have made their way out of the vasculature and can move into the actual injury sites where they play important roles for removing cellular debris, but also helping to modulate the remodeling response through release of different cytokines and chemokines. Now, this was the sort of the thought in most immune responses in response to injury was that most of these cells were uniquely derived from blood-derived sources or from the tissue itself. But during my postdoctoral work, a colleague of mine, Jing Wang, showed in a liver injury model, is that in addition to blood-derived cells such as monocytes that can differentiate into healing macrophage in the liver, is there's also macrophages that live in the serosal cavities that surround both your internal and thoracic organs. And in this case, macrophages that live within the peritoneal cavity have also the ability to migrate into the tissue from the outside in and are actually critical for that healing process. It's to note that these macrophages have a specific transcriptional profile and are often regarded as these GATA6 positive cavity macrophages. So this is a main transcription factor, which I'll refer to in the context of the pericardium. Now, the heart is also surrounded by a serosal cavity, the pericardial cavity, which has long been known to have important mechanical and structural roles and lubricating roles for the heart in normal functioning. And if you look at some older papers, there's been a number of reports of immune cells being present within pericardial fluid of patient samples, as well as growth factors and other cytokines that may play a role in sort of communicating with the rest of the heart. And the idea was to really explore its potential role in this process. However, in the traditional rodent model that Dr. Verzanten just presented, where you ligate the left anterior descending coronary of, in this case, the mouse heart, is this procedure also involves disrupting the whole pericardium. So essentially removing the factors in the cells that would normally live within this space and may affect heart function. So with the help of Dr. Daryl Belke and our cardiovascular physiology core in the Libin Institute, we actually developed a modified version of this coronary ligation system to first test, does the pericardium have a role to play in cardiac remodeling in maintaining cardiac function post-MI? And what our initial preliminary experiments showed that on both diastolic and systolic cardiac function measurements, is that keeping an intact pericardium actually has a dramatic benefit to maintaining cardiac function post-MI. And so this is 28 days post-MI, and this is compared to the traditional disrupted pericardium model. So this gave us the idea that the pericardium is in fact important for this process. And so we wanted to dive in deeper into what was regulating this. And so the first thing we looked at is whether there was immune cells present within the space. And in fact, there were. So this is profiles of the mouse pericardial fluid. There's a number of immune cells that are present, but I'll bring your attention to these GATA6 macrophage that represent roughly about a third of all these cells. And so they're the predominant immune cell type found within this compartment within the mouse. I then also collaborated with a very talented cardiac resident, surgery resident within the FEDAC lab, Dr. Ali Fateh Hassanabad, to actually look at this same profiling in the context of human patients. And so we took human pericardial fluid from patients undergoing cardiac surgery, and we're actually able to find that the patients also had a multitude of immune cells, and most notably is that GATA6 macrophage also represented a third of all these cells, of all this immune component, which tells us that these GATA6 macrophages seem to be conserved across different mammalian species and likely play a very important role within this environment. We did some further analysis in our mouse system comparing these pericardial GATA6 macrophage to other cavity macrophage, so in the pleural and the peritoneal cavity, as well as in the heart macrophage. And what we noted is that these GATA6 macrophage in the pericardium resemble sort of their cousins in both the pleural and the peritoneal cavity, but are distinctly different transcriptionally to cardiac macrophages. So although they sit just adjacent to one another, they are in fact quite different and likely have very different roles. So the next step was to look at, so what happens to these cells post-MI? And so what we noticed very early was that soon after we induce the MI, we actually get a full disappearance of these cells from the pericardial space. And this is at days 3 and 7, and a rebound at day 28. Again, collaborating with Dr. Fateh Hassanabad, looking at patient samples where we stratified different patient groups based on the time of pericardial fluid collection post coronary event. And so we looked at the inflammatory phase, so within the first 3 days, the proliferative reparative phase between days 3 and 14, as well as the maturation phase. And interestingly, we saw a similar dynamic where compared to patients undergoing a valve repair or replacement surgery that had no coronary event, there's a dramatic drop in early events, which starts to go up soon after and rebounds almost fully past the day 14 time point. Again, suggesting that this disappearance we see in the mouse system is also observed within the clinical context. So we thought we were onto something. And so trying to track these cells, we then turned to using some imaging modalities. And so here's just a epicardial surface of the heart that we used with confocal imaging. And this is a sort of a global view of the outside of the heart under baseline conditions. And this is a zoomed in image. And so in this case, we've used a reporter mouse system where we can actually tag our macrophages of interest, these GATA6 macrophage, with a green fluorescent protein. And what you can see is under baseline conditions, there's very little interaction with the outside of the heart, but this interaction changes considerably after you induce this myocardial infarction. And you get a huge amount of accumulation of these cells onto the surface of the heart. And if you take a closer look, looking at cleared tissue, what you can see is these cells are not only found on this epicardial surface, but as we turn the image here, is that many of these cells have actually migrated past this epicardial layer and have started to move into the myocardial tissue. So indicating that they're relocating from the pericardial space into the myocardium. And so it's all fine that they're getting into the heart. The question is whether they actually have an impact on function. And so using a genetic model where we're able to delete about 75% of these pericardial macrophage and comparing it to their wild type controls, what we noted is that in the absence of these macrophages, one of the readouts that we saw with the intact versus disrupted pericardial models is a measure of cardiac stiffness. So this is end-diastolic pressure volume relationship. And so without these macrophages, your hearts actually become quite a bit stiffer. And this is associated with increased fibrosis, particularly in the healthy remote myocardium of these hearts. And so just to summarize, the studies that I've shown here is that we've been able to identify that the pericardial space is actually home to multiple immune cell types, including these GATA6 macrophage in both the mouse and the human context. And they have the ability to relocate to the heart following ischemic injury and play a role in mitigating some of the fibrotic responses in the heart. Now, some of the questions moving forward are really trying to understand the locations of where they have their role, so whether in the pericardial space or in the heart, and ultimately what type of mechanisms they use to actually regulate this response. And we think they're directly interacting with fibroblasts within those local environments, and we have ongoing studies to look at. But what I hope to impress upon you here is this idea that the pericardial space is an active sort of component that really contributes to heart remodeling, and may really be a space that is ripe for targeting therapeutically to further enhance or modulate these remodeling responses within the heart. And so with that, I'll pass it back on to Dr. Fidak and Vizantin to finish off just the last portion.
Dr. Dennis,
Yes.
Is it the end of the presentation?
It is, yes.
Okay, thank you so much. That was an excellent talk for all of you, the Prof. Fidak, Dr. Vasantan, and Dr. Dennis. I think there are many questions from the audience, but I think I will let Dr. Puspita to ask question first. And we have several experts from biomedical engineering as well, and also from Research Center of Biomedical Engineering, Pak Yudan could address a question. It was very interesting, all of the talks today. I wish that we could start research in Indonesia. We have several research centers, especially in university base. It is Universitas Indonesia, the biggest university in our country. We have medical research unit, and we also have biomedical engineering research center. So we can collaborate together, and we hope for collaboration with Dr. Fidak and team. I think Dr. Puspita or Pak Yudan want to ask question. If not yet, so I would like to ask Prof. Fidak. I would like to ask about the bioscaffold. What do you think about the source of bioscaffold from decellularized amniotic membrane? One of our cardiac surgeon start research about decellularized amniotic membrane. What do you think about that?
Yeah, that's a great question. So the source, and there's so many options for sources of biomaterials, like, whether it come from porcine or should we be using human biomaterials? Which tissue should we use? And I think what's interesting is there are some tissues that are highly regenerative, right? Like the intestinal lining, or, an amniotic or a placental tissue could be much more regenerative. And we could get a better response from some of those tissues. So to be honest, we don't know what the optimal biomaterial is. And I think that, amnion or placenta, we're actually exploring placenta right now in the lab as an alternative. And I think it's going to be really exciting to see which ECM from what tissue will work the best. And I suspect that, we're going to see that certain tissues that are more regenerative will probably give us a more robust response. So I think that work is very important.
Yes, we just, I think this group is Dr. Arza from cardiac surgeon. He started research for his PhD research with big decellularized amniotic membrane provided. Maybe he could contact you to ask about how that this possibility of the bioscaffold will work.
Absolutely. And in fact, I think I might have seen that work before as a reviewer. So it sounds familiar, and I think it did come from Indonesia. So I may be familiar with that work, but yeah, I think it's great. And nice to see another cardiac surgeon doing this type of work. Fantastic.
Thank you for your kindness. I think I saw the question for Dr. Puspita. Dr. Puspita, you can answer the question.
Sorry, what was the question again?
Yes, I think this question is from Giska Ramodita.
Oh, okay. Current technology of IPS cardiomyocyte in Indonesia. So actually that's what we've been doing to develop with Dr. Winda at IMERI. So it's still under development. For other lab, we don't know for sure, but I believe there are some labs too doing the development of IPS because currently most of the lab in Indonesia is focusing on adult mesenchymal stem cells. Because before, like pluripotent is mostly on embryonic stem cells, and in here due to the ethical reasons, so that's why we focus on adult mesenchymal. But due to the reason of development of IPS, because of the technology, but because because of the reagent itself is, at that time it was quite expensive to us, and because there are then we had to wait for several months, so maybe that's one of the reason why it got delayed. But for the time course when it will be applied, I can't say for sure, because we still have to, for the lab environment itself, we still have to do optimization as well. So maybe within 5 years or more, I think that would be ideally realistic for Indonesia. I hope it helps answer the question.
Yes, thank you, Dr. Puspita. I think Prof. Vedak, Dr. Vasanton, and Dr. Dennis said we've just started the research for cardiomyocyte from human iPSC. But it's just started, so we have a difficulty in especially in the culture environment in our lab in Indonesia. The humidity, because Dr. Puspita have done it at Nottingham, so it's very different in Jakarta.
Right, interesting. Yeah.
Can I ask a quick question to Dr. Puspita?
Yes, please.
I just had, so the heart is made up, as Paul mentioned, is made up of sort of a multiple cell types, including fibroblasts, endothelial cells, immune cells. So where are we in terms of iPSC cells, but in combination with some of these other cell types to sort of get a multicellular sort of scaffold to work with and to test these? If you can just comment on that quick.
Yeah. So actually, like before I came back to Indonesia, like we've been trying to do like a co-culture system when we had a collaboration with universities I mentioned before. But because we found difficulties in actually bringing that for the drug screening, so we postpone it. So we only focus primarily on cardiomyocytes. As for the AHT itself, it's a combination of cardiomyocyte and fibroblasts. And for Indonesia, of course, like we are still trying to do the first step. So we are focusing on doing the cardiomyocytes first before we can actually can do more like some kind of more complicated like co-culture systems. But it's actually good because in the in vivo settings, it's not only cardiomyocyte, of course, we also get endothelial cells, smooth muscles, and then fibroblasts as well. Yeah.
All right, thank you. And I think there is one question for you, Dr. Vasantan, in the chat box about the immunostaining of alpha SMA. Did the implantation live also induce activation of triiodothrionin maybe? Oh no, I don't understand. Fibroblast to myofibroblast?
I can help with that. Yeah,
Okay.
I saw that you showed some cool images from the confocal that you showed the alpha SMA staining. So my question is, did you see that from the implantations of 3 days versus 7 days, did you see the more activations of the myofibroblast, like more fibrosis or like less fibrosis as in by the Dr. Fedak's introduction?
Yeah, thanks very much for the excellent question. So in terms of the activation of myofibroblast and staining, we do have fibroblast changing phenotype data. Specifically in that image I showed, we mainly used alpha SMA as a method to highlight medium to large size blood vessels in that case. So that was what we used it for in that imaging. However, in terms of looking at the conversion of a myofibroblast-like phenotype and the fibroblast-like phenotype, we found that the Cis Ecm biomaterial does change it, and that's based on morphology assessment and also collagen gel contraction. So what we see, and it's likely through fibroblast growth factor 2, that molecule interacts with the fibroblasts, and it lessens the myofibroblast activity and switches things more into a paracrine fibroblast that is less fibrotic.
Okay, cool. And if I can ask you more questions on that, like in insured tissue, is it usually we have more cardiomyocytes than the fibroblasts, or like how it usually, and maybe also related to Dr. Denisat's research, like what is usually the percentage of cardiomyocytes, fibroblasts, and macrophages in injured tissue?
So I can answer mainly for the fibroblast section, and it really does depend on where you read. The fibroblast is one of the majority cells in the heart, and it regulates many of the processes, even though, say, the cardiomyocyte is the most advertised. So depending on where I've read in review articles and textbooks, sometimes I've seen as low as 40%, but as high as 75%. And the fibroblast is one of these majority cells that is regulating the processes around the myocyte to help with that extracellular matrix regulation. So that would be the percentage I can quote for the fibroblast.
Well, maybe just add on. Oh, sorry, go ahead, Paul.
Yeah, I was just going to add it. I think what happens is, I mean, the fibroblasts and non-cardiomyocyte cells in the heart get underappreciated because they're very small. The myocytes take up all the space. It's like, you know, Canada, it's a big country with very few people. And Indonesia has, you know, lots of people in a smaller space. So, you know, we believe that the fibroblasts are really important, and there are many of them. And of course, with injury, there's lots more, right, around the injury site. And then Justin will tell you more about the immune cells.
Yeah, so under baseline conditions, you sort of have, fibroblasts have sort of gained more recognition for their density within the heart. But immune cells, the heart is also chock full of immune cells, notably cardiac macrophages that interdigitate between the cardiomyocytes. In the context of the infarct, so in the infarcted tissue, so Dr. Vizanthan showed the image of the fibroblasts sort of lining up or butting up against the biomaterial. So the infarcted region is chock full of these fibroblasts and myofibroblasts. But if you were to stain immune cells on top of that, they would be juxtaposed one on top of another. And so you have a lot of death of your cardiomyocytes, but it's replaced by infiltrating cells or migrating cells, such as the fibroblasts, to sort of fill in the gap and to start forming the scar. Keep in mind, the model that we're using is a permanent occlusion model, so we're getting quite a lot of cardiomyocyte death. If you were to use an ischemia reperfusion model where you get a more modest loss of cardiomyocytes, you may see a more different distribution. So it's really model dependent, and it's also the technique that you use to measure that you'll get different percentages. But you definitely have, in the infarct itself, you have a lot of fibroblasts, myofibroblasts, as well as different types of immune cells.
Okay, great. Thank you. I think I will let another participant to ask question. Okay, I think it's from Pak Yudan. Pak Yudan is the Director of Biomedical Engineering Research Center. For Dr. Vasantan, please.
Sounds good. So to reiterate the question, it's about the physical parameters of the Cis Ecm and methods of fabrication. So to highlight this biomaterial, it's a commercially available biomaterial. So many of the design specifications are in fact proprietary from the original company, and it's under the trade name Core Matrix Cardiovascular. In terms of basic details for these physical parameters, the Cis Ecm, so the small intestinal extracellular matrix from a porcine source, is stacked in 4-ply. So each ply, each individual ECM is stacked one on top of another. The way it's prepared, there is kind of minimal processing after the decellularization. And the importance there is that it can keep many of the bioactive factors like fibroblast growth factor 2. The main preparation for there is lyophilization, which is essentially freeze drying. So after you stack the material together and freeze dry it, that's the bioactive material we work with. I hope that answers in terms of geometry. So there's 4-ply, and that helps the cells kind of interdigitate within the material. And then in terms of fabrication as well.
Okay, thank you, Dr. Vasantan. I think we're almost running out of time, but as a pediatric cardiologist, I want to raise question because Prof. Vedak, maybe for pediatric population, we have, of course, difficulty treating dilated cardiomyopathy. The peak incidence in infant less than 1 year, but they already have a huge left ventricle. And in Indonesia, we still have no option for fat ventricular assist device or even heart transplant. So we give the medications like captopril, furosemide, and digoxin. But I think we have to think about the future of this baby, because the heart is very huge. This is not disease specific like Pompe disease, but maybe for from viral myocarditis and continue to become a dilated cardiomyopathy. I think we must start a research for this subgroup. What do you think?
Yeah, absolutely. And one of the things, we focus on ischemic heart disease because we deal with adults mostly, but the mechanisms of reducing fibrosis and altering inflammation are just as important for non-ischemic dilated cardiomyopathy. And we don't have a lot of treatments for those patients, as you know. But interestingly, the treatments that do work, like, ACE inhibitors and spironolactone and things like this in these patients likely work through fibrosis pathways. So if we can better target fibrosis pathways in dilated cardiomyopathy, we may be able to prevent the progressive ventricular dilatation and thinning. and the cell death that leads to the decompensated stage. And in fact, our work, so we've tried to make our work more applicable to more patients, including pediatric populations, by taking our biomaterial and micronizing it, and we can now inject it. So what we've been doing, and we didn't have time to show you all that data, but we've been injecting that micronized matrix into the pericardial space, and Dr. Denisette has been showing that it's altering inflammatory pathways. So it's an opportunity. So even babies, you could do a pericardial puncture, inject the material into that space, and probably affect the entire, in a dilated cardiomyopathy, have a way to affect the entire ventricle and alter the fibrosis and the inflammatory pathways. So we're very excited about work like that. And we're hoping actually to try to get this to clinical trials within about 18 months. And to be honest with you, we're hoping that we can find some partners outside the country to maybe help us with some of those trials. So it may be, you know, your group and your hospital could be interested in that down the road. We'd be happy to chat with you more about it.
I think that's a good news. I work with my colleagues from Pediatric Cardiologist Society in Indonesia. That the pediatric cardiomyopathy is very hard to do. I mean, as a pediatric cardiologist, with some kind of frustration, because we found the case since baby, maybe 4 months old or less than 1 year, but maybe the remodeling is ongoing, and then we have no other option to treat them. Okay, hopefully we can continue for next meeting.
Yeah.
Yes, thank you so much. It's all of you, thank you so much for excellent talk. Prof. Dr. Fasantan, Dr. Denisette, and also Dr. Puspita, thank you so much. And for all participants from our colleagues from all over the country, and also from Biomedical Engineering and Faculty of Engineering Universitas Indonesia, we would like to thanks for all your participation, and we hope for further collaboration in Indonesia, and also hopefully with Dr. Fidak and team from Libin Cardiovascular Institute. So I would like to close this meeting. Thank you so much, and I will give it back to our host.
Thank you so much, Dr. Winda. Thank you again to Dr. Paul Fidak, Dr. Vishnu Vasantan, and Dr. Justin Denisette, also Dr. Puspita, for your time and for sharing with us all your researches. Surely the lot of us here will find your insights extremely helpful, especially some of us being students hoping to pursue a career in STEM. And as we're nearing the end of today's guest lecture, let us commemorate this event with a photo session. So to the rest of the participants, please open your camera to take the picture, and to the committee, you can start counting down to screenshot.
Smile.
To the committee.
Okay, finish. All right, I think. Okay, thank you so much.
Thank you very much. Nice to meet everybody.
Yes,
Yeah, thank
nice
you very
to meet
much.
all of you.
Thanks so much.
Looking forward to see you again.
Thank you.
Yeah,
Bye bye.
absolutely. Take care. Bye.
Thank you.
Thank
Have a great
you.
day.
Okay, I think we can close this meeting. Terima kasih kepada semua yang sudah hadir, kepada panitia yang sudah mempersiapkan acara ini dengan sebaik-baiknya. Kita akan tutup acara ini, nanti akan kita kumpulkan daftar peserta dan akan kita kirim e-certificate dalam waktu 2 minggu, sekitar 2 minggu.
Terima kasih.
Oke, terima kasih.
Terima kasih.
Ya, bisa jingkelnya mungkin.
Wahai perempuan Indonesia, tercipta sebagai ibu bangsa, melahirkan insan yang mulia, sehat dalam jiwa dan raga. Kami terdepan dalam tugas untuk melayani kesehatan Anda, melindungi sang buah hati agar sehat senantiasa selamanya. Rumah sakit anak dan bunda, harapan kita.
Terima kasih teman-teman dari kardiologi anak, Dokter Tina, saya lihat Dokter Yosi, Dokter Rizky, Dokter Supraheta. Semoga bermanfaat acara kali ini dan bisa kita lanjutkan di acara nanti guest lecture lagi, dan mudah-mudahan ada kerjasama dengan Prof. Fedak ya untuk bayi-bayi di Indonesia dengan kardiomiopati. Terima kasih.
Dr. Winda, saya pamit leave ya.
Jangan buru-buru nih. Oke, Dokter Puspita, nanti kita diskusi-diskusi lagi. Mungkin ada yang saya juga nggak terlalu nangkep nih. Mungkin perlu penjelasan lagi nih dari Dokter Puspita nih.
Saya juga sebenarnya tadi udah agak ngantuk.
Kenapa? Itu ya, nyiapin slide ya.
Jadi kan saya hari Rabu kemarin kan booster AstraZeneca.
Oh.
Terus ada kipinya lumayan banget.
Ya ampun.
Lemes, sakit kepala sama menggigil
Meriang
gitu
ya?
loh. Iya. Jadi
Ya
baru sebenarnya kemarin malam.
Waduh, untung sih ya, alhamdulillah ya bisa tampil ya, Dokter Puspita. Cepat sembuh lagi nih, semoga sehat terus.
Iya, makasih dok.
Ya, terima kasih. Silahkan kalau mau live dulu teman-teman semua juga. Dokter Puspita terima kasih.
Ya, izin live.
Ya, terima kasih. Ya, terima kasih ya panitia ASB. Hebat ya teman-teman dari ASB sudah bekerja keras. Teman-teman dari Diklit juga. Baiklah, kita tutup saja Zoom ini. Sampai jumpa lagi ya di guest lecture selanjutnya.
Makasih, Dok.
Terima kasih Bu Dokter.
Ya, Aldo, ya,
Terima kasih Dok.
Siva semua. Oke.
Ita. Pagi, Ita.
Dok, Dokter Tina.
Aduh,
Hostnya masih belum matiin. Ayo, dimatiin.
iya, kira pengen ngomong deh. Ngomong sama Ita tadi.
Maaf ya dok, ini kita lagi ngobrol-ngobrol berkadang ini
Iya.
YouTube-nya juga belum mati.
Oh.
Dokter Tina, hai. Hai.
Aduh.
Maaf ya,
Tenangnya.
omongannya nggak jelas dok, saya nggak paham.
Iya, iya. Tapi bagus loh. Yang dokter apa itu kan bagus, yang myocardial.
Iya, makanya dok, nanti kita undang lagi dok. Yang
Iya.
mana dok? Yang mana dok? Yang India?
Iya, enak kalau banyak apa operasi miositnya kan bisa diambil.
Iya.
Iya.
Aduh.
Iya,
Saya nggak
iya.
paham dok ya saya.
Pintu kali, oke.
Makasih ya.
Sampai ketemu ya.
Ya, kita screenshot nih ke sini. Screenshot ya.
Iya, iya.
Sana dikit. Halo. Ya, satu, dua, tiga, screenshot. Oke dok.
Oke, ya. Bye-bye.
Bye.
Ya,
Medan, hore Medan.
Ya. Medan, ya, ya.
Ya,
Live ya,
dok.
izin live ya.
Dok. Ya dok. Rajin Dokter Tina.
Ya, iya.
Oke. Salam sama abang. Denny. Yoshi. Ini belum ada ditutup sih. Yoshi udah lagi di mana dia? Tinggal 7 orang, ada Yoshi. Kayaknya Yoshi. Oh, udah ditutup. Belum. Yoshi. Ayu nih nggak nutup ini, dia sambil nih Ayu nih. Yuk Yoshi. Ayo sibuk ngajar tapi HP dihidupin. Kesian itu HP. Matiin. Ayo. Yoshi. Ayo. Oke. Lip lah ini. Ini kenapa nggak ditutup nih orang-orang? Zoom-nya belum ditutup ya.